So I think it's been a fantastic afternoon. I'm not a clinician, I don't see patients as Michelle does, but I come to events like this and I just feel the energy that Michelle has generated from this cohort. The 1,000 patients and their relatives that we study in the clinic, many of them come along to events like this, and I'm just struck by the effort that they've put in to support our work. And I just think it's a really exciting atmosphere and environment for us to discuss both the clinical work and the science. It's often difficult to get over the exact details of the science we do, but we try and put over the energy and the enthusiasm and the efforts that we're making. And I find these afternoons tremendously rewarding to chat to the patients and to have them hear what we're up to.
So the focus of my research has been mainly looking at some of the current medications that we have available in Parkinson's. And what I'm talking about today is a side effect of one of those medications called impulse control behaviors. It's very important to understand these kind of effects because it can direct not only how we use these medications in patients at the moment, but also it tells us more about the underlying processes in Parkinson's and help look for other treatments in the future.
We know that sleep's important for day-to-day activities, including things like memory and attention. And I'm really excited to be working on the Wearables project to look at using wearable devices to study sleep in detail in RBD and PD.
If a patient were to use our device post-testing, we believe that it will improve their quality of life. It'll help them to walk more freely, to live more independently, and to become more active. A lot of patients that have a higher fall risk, they suffer from repeated falls, often withdraw from everyday activities, which is not good for them, not good for quality of life, not good for their disease prognosis. So our device should help these patients to become more active and more independent.
What we're trying to do is look at people before they get Parkinson's. So we're studying a group of patients who have a condition called REM sleep behaviour disorder. So this is a condition where people move in response to their dream content, and so they can therefore display quite violent behaviours sometimes during their sleep in response to their dreams. And people who have this disorder have a very high risk of developing Parkinson's in future. So that means by studying them, when they don't have Parkinson's, we might be able to look at the very early stages of how Parkinson's disease starts and spreads.
So microglia are the vacuum cleaners of the brain, and so they are phagocytic cells that go around eating all of the debris that is produced of a normal day in the workings of your brain. And so they're really incredibly important in just keeping the brain nice and clean and tidy under normal circumstances. And we think that when you get diseases such as Parkinson's disease, that they lose their capability to be able to keep everything nice and tidy.
Some of the key scientific discoveries that we've made over the last few years, and we tried to describe this afternoon, focus on this use of patient stem cells to study Parkinson's. So because of the generosity of the patients that we work with, we're able to obtain skin biopsies from those people. We can take them back into the laboratory, reprogram those skin cells into stem cells, and once you've got stem cells, you can make any cell type you're interested in. So for a long time, the work of the Parkinson Centre has been to make dopamine neurons, these key neuronal cell types that start to die off in Parkinson's. We've made these neurons from patients, from healthy individuals, and identified some of the changes in cell biology that occur in these neurons. For example, around protein stress, handling protein, folding protein. And if proteins such as alpha-synuclein can't be folded correctly, the cell simply ejects them out of the cell. And this idea of neurons throwing out alpha-synuclein happens in the dish that we study, it happens in the brain as well, may be one way in which Parkinson spreads throughout the brain.
I think in terms of where we can really start though, and where we actually talk about today, is in drug repurposing. So these drugs are, examples would be gabapentin, pregabalin, trazodone, which have already been through this expensive regulatory, time-consuming 10-year process and actually have symptomatic benefit for many patients with Parkinson's in sleep, for example, pain and other non-motor symptoms, anxiety and depression that people suffer. And so for me as a clinician, it's almost a win-win because people are very happy to take this if they're not sleeping well, and you can say it will help your sleep, but if you can also say it might slow down the progression of your Parkinson's and where you're going to be in 5 or 10 years, I mean, they're delighted to take it. Obviously, gathering the clinical trial data that we need will be obviously quite a long and expensive process, but it's absolutely necessary. And I think the other thing for clinical cohorts is really having the right tools. So having the right types of biomarkers, the right types of wrist wearables, clinical markers, blood-based biomarkers, sleep markers, really from the patient's own home environment to the clinic, to the bed, to the bench side is absolutely critical.